All right, thanks so much. So today I'll be sharing some of the work we've been doing over the last few years, focusing on the transmission of foot and mouth disease in wildlife-livestock interfaces in Kenya. Sorry. Okay, so one of the chief challenges for the control of FMD in the African continent is the presence of a wildlife reservoir, the African buffalo. Most of what we understand about the ecology of what's going on at this interface between wild animals and domestic livestock has really been largely done in southern Africa. And southern Africa has a very different epidemiological context in terms of what's going on with the wildlife than what we see in eastern Africa. So in East Africa, we see kind of a hyper-endemic state of FMD in the cattle population, and we also see very integrated interfaces. We don't have a situation where the wildlife are fenced or only in national parks. We have these sort of landscapes that are very much shared and used both by the wildlife and the livestock. Yet we don't really know what the role of the buffalo is in these sorts of landscapes in East Africa. In Kenya, what we do know is that the seroprevalence in the buffalo populations is upwards of 65%. That's pretty typical of what we see across Africa and buffalo. Seroprevalence in other wild ungulates is quite low, so we're not really sure on what the role of the other species may be. And in Kenya, the national seroprevalence for cattle is over 50%. And one of the reasons we can't really resolve the degree to which the buffalo and the cattle are sharing FMD is that we have very little molecular data or sequences from the buffalo populations in East Africa. I think prior to us starting this study, there was only two sequences available in buffalo from Kenya, and I think there was only a handful for all of East Africa. So our objective was to sample and characterize the foot and mouth disease currently circulating in Kenya, characterize the frequency and directionality of transmission between buffalo and cattle, and then to describe the between-herd transmission dynamics in the buffalo population. We conducted the study in Olpejeta Conservancy, which is located in central Kenya. Olpejeta is about a 350 square kilometer wildlife area, but importantly, it's also a commercial cattle ranch. So we have about 1,200 buffalo in this ecosystem and around 5,000 cattle. And this picture I show up on the right here is an example of a water trough that's been constructed for cattle, and you see that the buffalo are happily using that and even wading in it. So there's definitely, if anywhere, there would be abundant opportunities that we would think that transmission could occur in this sort of ecosystem. Importantly, I put this picture of these rhinos here. This ecosystem is fenced, so we have kind of an intact ecosystem where we have intermixed wildlife and livestock within a fenced situation, and that's primarily due to the security required to protect rhinos from poaching. So essentially we have these cattle that are intermixed with the buffalo, and they are relatively isolated from the cattle in the surrounding communities. And the Olpejeta cattle population did experience a clinical FMD outbreak in 2013. So we sampled 92 buffalo from the Olpejeta ecosystem. The seroprevalence in these animals was around 77%, and we were able to recover 80 VP1 sequences. 30 of these were SAT1s, and 50 of these were SAT2 sequences. And we had 6 individual animals that were dually infected by both. For the cattle, we collected 98 samples, and we also had available to us 21 outbreak samples from basically from central Kenya. And the seropositivity rate in these animals was 92%. And then we were able to get in total from the cattle samples that we had 17 VP1 sequences. From the Olpejeta cattle, we were able to get 4 sequences. 3 of these were from the probangs that we collected, and those were all serotype O. And then the outbreak sample that we had from that 23rd outbreak was from serotype A. I just put this figure up just to kind of set up the next analyses that we performed. These are maximum likelihood trees for SAT1 and SAT2 for all of the VP1 sequences available in sub-Saharan Africa. And I'm highlighting the upper areas that are in color. The blue sequences are our new sequences. The red sequences are all from GenBank. And the clades that I've highlighted in blue and red are the clades that we're going to be focusing on for the subsequent analyses. We didn't do this for the full continent. We really focused on these clades, which include the currently circulating SAT1 and SAT2 viruses that have been found in Kenya, not just by our study, but by other recent studies. So we performed ancestral state reconstruction with a Bayesian framework with BEAST. So these are what these trees look like. Now we have the blue representing cattle samples, the red representing the buffalo. And basically, this sort of analysis allows us to try to infer what we think the ancestral population is for the circulating viruses that we find. And then it also tells us something about how frequently we think that transmission occurs in either direction between these two host species. And for both SAT1 and SAT2, the evidence strongly suggests that the cattle population is the population of origin. So we see that for both SAT1 and SAT2, but what we also notice is that the populations, or the viral populations, are very much intermixed. We don't see a separate population of viruses that are found in buffalo, which has been previously suggested, but this is not what we see here. I also want to emphasize that this is for the currently circulating clade. So this is not saying that overall SAT1s and SAT2s have a cattle ancestor, or that's the original host. This is only talking about these currently circulating clades of SAT1 and SAT2 that are in East Africa. This is the same tree looked at a slightly different way. Now we've colored the branches in blue if they're buffalo, and then the black are the cattle. And because we were able to recover so many sequences from buffalo, we had a unique opportunity to start looking at between herd transmission dynamics for these buffalo. So the buffalo are in the blue branches, and now we have colored the tip labels according to the herd that was sampled. So on the day of sampling, which animals were in the same herd. So it's kind of a snapshot of the host population structure. And what we find from an initial examination of this tree is that we have this clustering that appears where we have based on the occurrence of this river that bisects the ecosystem, we have clustering in the tree that seems to be related to this fine scale geographic features. So we have FMD from herds that are mostly on the west side clustered together, as well as on the east side. Now, we wanted to look at this further, so we looked at a metric called phylogenetic divergence, which essentially measures or asks the question, do different herds have distinct populations of foot and mouth disease? And we do this by measuring the mean pairwise phylogenetic distance between sequences that are found in these different herds. And then we test the significance of that measure with a permutation test with the null hypothesis that there's no phylogenetic clustering between herds. So this is what these output look like. So each of these panels represents these comparisons between different pairs of herds. The vertical red line represents the observed value for this phylogenetic distance between these two herds, and the curve represents the null expectations when we do these permutations. And in all of these cases, for these three herds, which we had sufficient power to look at this, we find that these herds have significantly different populations of foot and mouth disease. And two of these herds are actually were sampled both in the area that's labeled low Drew. It's a very small area, and that suggests first that the herds that the way we are doing this snapshot on the day of sampling does seem to represent something significant about the host social structure, and also that there may be limited opportunities for between herd transmission. And I'd also like to emphasize, too, that the scale, if you look at the scale at the bottom, we're talking about very fine scale here. This is less than 5 kilometers between these different herds. So we did the same thing for SAT2. Immediately on a visual scan, we don't see as apparent of clustering for these herds, and we repeated the same analysis, and we had more data in this case, so we have more herds that we were able to do this for. But what I'll highlight is, first of all, we had 3 herds that actually showed less divergence than you would expect randomly, which suggests in these cases, these are different herds than the previous analysis. These 3 herds may not actually be distinct epidemiological units. They may intermix. They are found in this kind of southeastern area, so these don't seem to be distinct herds. But we do find one herd that does show a very different pattern, and this herd was found very close to the other herds, and this is located, this lower cluster here is this herd that seems to be an outlier, and they have a very unique virus population that's found only in that herd, and that herd also doesn't tend to have any of the other viruses found in the rest of the area. And we're talking here, this distinctive herd here is only about 2 kilometers from the others, and there's actually no river separating them. So it's interesting to see such fine scale structure in the viral population based on very small differences, or sorry, fine scale spatial differences in this population. Okay, so our main conclusions are that it appears that the SAT1 and SAT2 viral populations in buffalo cattle are intermixed in Kenya, and that the current circulating clades of SAT1 and SAT2 appear to be of cattle origin. Second, buffalo to cattle transmission seems to be rare, even in shared landscapes, and we're basing this on this old Pejeta cattle population. Even though they are in close contact with the buffalo, we are not able to recover any SAT1s and SAT2s, and instead the only serotypes O and A have been found in this particular population. And third, transmission in buffalo is influenced by fine-scale social and/or spatial structure in this population. So some limitations that we have to this study. We were thrilled to be able to recover 80 sequences from one population, but these sequences were all collected at a single time point from this single population. So, and then compare that to the rest of Kenya, or even more broadly East Africa, where we have very few sequences. So very densely sampled population, which limits how much we can say broadly about what's going on at the wildlife-livestock interface in East Africa. We can say a lot about this particular population, but we would need to do more sampling in other buffalo populations to make broader conclusions about the frequency with which this sort of transmission is happening. And also a lot of the conclusions we're making about the herd structure, this is actually not a population that's monitored. This is basically a snapshot of the social structure on a single day. It did suggest, or the results suggest that this was a meaningful way to do this, but certainly actually monitoring that buffalo population would allow for much better insights. So some of the next steps that we're working on is to model the spread of FMD in these sorts of systems. One initial piece of work we did is in the same ecosystem in Old Pejeta. We placed, or our collaborators placed, GPS tracking collars on every single herd of cattle, and then tracked how these herds were using the landscape. And from that we can infer where contact has occurred and build a dynamic contact network. And once we have a dynamic contact network, we can simulate the spread of an FMD-like disease through these sorts of networks based on contact. And we found that just only looking at the contact network and no other types of seasonal changes, we get outbreaks that are significantly larger, about 10% larger in the dry season than the wet season. And that's just purely based on the changes in the contact structure, higher contact rates in the dry season, clustered mostly around the water sources. But this model was very simplistic, and we did not incorporate any wildlife data here. So some of the ongoing work we're doing now is we have placed these GPS tracking collars on buffalo, as well as the impala, which is hypothesized in southern Africa to be a key bridge host of how FMD jumps into cattle populations. So this is in a different ecosystem. It's in southern Kenya and the Masai Mara. And so right now this is still actively going on, but we're collecting data on the space use of these species. At the same time, we have collected data on the movement of cattle in this ecosystem. So I'm not sure why these dots are supposed to be connected by lines, and it's not appearing on this screen, so sorry about that. But we basically collected data on where these different households that are part of different villages, where they're seasonally moving their animals to pasture, and we can build a network from that and overlay that on our our wildlife space use behavior to get a much more holistic picture of what's going on in this ecosystem with the wildlife and the livestock. So that's kind of where we're moving on some of this work. Just want to take a moment here at the end to acknowledge the large team that made this project possible, including Kenya Wildlife Service, which is our key partner in Kenya, as well as the Kenya FMD lab, and then the USDA at Plum Island did all of our lab work, and we're very grateful to them. With that, I'd be happy to answer any questions if there's time, of course.